For decades, traditional computing has been built upon the foundation of silicon-based processors, transistors, and binary logic, an approach that has powered the digital revolution, artificial intelligence, and the internet. However, as Moore's law slows down and the demand for faster, more energy-efficient, and exponentially powerful computational systems skyrockets, researchers are turning to an unexpected and revolutionary alternative: biocomputers powered by DNA, proteins, and molecular structures. Unlike classical computing, which relies on binary zeros and ones, biocomputing leverages the natural properties of biological molecules, allowing information to be stored, processed, and executed in ways never before imagined. DNA, the fundamental building block of life, holds the key to a new paradigm of computation, one where data storage can be achieved at densities millions of times greater than today's best semiconductor technology, and where computations can occur at the molecular level, enabling breakthroughs in fields like artificial intelligence, cryptography, and medicine. But how exactly does DNA computing work? And more importantly, what does it mean for the future of technology and humanity as a whole? At its core, DNA computing harnesses the natural processes of DNA replication, hybridization, and enzymatic reactions to encode, manipulate, and retrieve information. Traditional computers perform calculations through electrical signals passing through silicon chips. But in a DNA-based system, computations occur within test tubes or biological environments where DNA strands are carefully designed to interact in predefined ways, solving complex mathematical problems in a massively parallel fashion. One of the most astonishing aspects of this approach is its efficiency. A single drop of liquid containing DNA can perform trillions of computations simultaneously, something no supercomputer on Earth can achieve today. This is because DNA strands naturally seek complementary sequences, a property that can be used to execute logic operations, solve optimization problems, and even create biological circuits that function similarly to digital circuits, but in a much more compact and energy-efficient manner. Unlike conventional processors that require vast amounts of electricity and generate significant heat, DNA computing operates at room temperature with minimal energy requirements, making it an environmentally friendly alternative that could revolutionize data centers and high-performance computing. One of the most exciting applications of biocomputing is data storage. As the world generates an overwhelming amount of digital information, currently estimated at over 2.5 quintillion bytes per day, traditional storage solutions like hard drives and SSDs are struggling to keep up. DNA, however, offers an elegant and nearly infinite solution. A single gram of DNA can store over 200 petabytes of data, and it remains stable for thousands of years, unlike magnetic or optical storage, which degrades over time. Scientists have already demonstrated the ability to encode entire books, images, and even short films into DNA molecules, retrieving them with perfect accuracy. This has led major technology companies, including Microsoft and IBM, to invest heavily in DNA storage research, envisioning a future where massive data centers could fit inside a small vial of liquid instead of sprawling warehouses consuming gigawatts of energy. The implications of this are staggering. Not only could it completely change the landscape of cloud computing, but it could also enable entirely new forms of digital preservation, ensuring that critical knowledge and history are stored in a virtually indestructible format. Beyond storage, biocomputers are set to revolutionize medicine, bioengineering, and artificial intelligence. One of the most promising developments is the use of DNA-based nanorobots that can function inside the human body to detect diseases, deliver targeted drugs, and even repair damaged cells. Imagine a future where instead of taking a pill, you could receive an injection of programmable DNA nanomachines designed to attack cancer cells with precision while leaving healthy cells unharmed. Researchers are already developing DNA circuits that can act as biological decision makers, determining whether a cell should undergo self-repair or apoptosis, programmed cell death, which could lead to breakthroughs in curing previously untreatable diseases. Moreover, DNA-based computing could lead to the creation of biological artificial intelligence, AI models that are not only inspired by nature but actually run on biological substrates. Instead of neural networks being simulated on silicon chips, future AI could be built directly within biological systems, enabling machines that learn and adapt in ways far beyond our current capabilities. But with such an immense technological leap come profound ethical, security, and societal concerns. How do we regulate biocomputational technology to prevent misuse? If DNA-based storage and processing become widespread, will we see the emergence of new forms of hacking, where biological data can be altered or corrupted by malicious actors? Could there be risks of biohacking, where DNA computers are used to create harmful biological agents? These concerns are not science fiction. They are very real and require urgent discussions about governance, security, and ethical responsibility. Governments and research institutions must establish strict protocols to prevent the potential dangers of DNA computing from being exploited. Another pressing question is whether the integration of biology and technology will blur the boundaries between living organisms and machines. Could we eventually reach a point where DNA-based AI merges with human biology, creating a new form of hybrid intelligence? And if so, what are the implications for our understanding of consciousness, identity, and autonomy? Despite these challenges, the potential of biocomputers is too groundbreaking to ignore. Researchers around the world are making rapid progress, with DNA-based logic gates, storage systems, and even programmable biological circuits already being demonstrated in laboratories. The next decade could see the first large-scale implementations of DNA computing in real-world applications, leading to unimaginably powerful new forms of computation that far surpass anything silicon can offer. In the coming years, we may witness the emergence of hybrid systems, where traditional computing is augmented with DNA-based processors, combining the best of both worlds to achieve new heights in efficiency and capability. Companies, governments, and scientific institutions must prepare for this transformation, investing in research, security, and regulatory frameworks to ensure that DNA computing develops in a way that benefits all of humanity. So as we stand at the brink of this biocomputer revolution, one thing is clear. The fusion of biology and technology is no longer a distant dream. It is happening now. The question isn't whether DNA computing will change the world, but how soon we will fully harness its power. Will we witness the rise of biological supercomputers in the next decade? How will DNA storage reshape the way we archive and access information? And ultimately, how will these advancements redefine what it means to be human in an age where computation is no longer confined to silicon chips, but flows through the very molecules of life itself? The future is being rewritten at the molecular level, and we are only just beginning to unlock its true potential.